Hi everyone, I'm Susanna. Today I'm going to introduce my studies about goals and their impact on host plants' reproductivity. I hope through this video you will gain a better understanding of how parasitic organisms like goals can influence plant growth and their reproductivity. Have you ever seen the gulls? Many people might think that the round objects they see on branches are fruits, but they are actually gulls. Gulls are abnormal plant growth induced by insects. They are commonly found on branches near seeds, and they extract nutrients from the host plant to sustain the growth of the larvae. If we use a more metaphorical description, gulls and host plants are like uninvited tenants and landlords. These tenants consume the limited resources within the house, which naturally affects the landlord's well-being. If we continue with this analogy, our research is like investigating how the landlord reacts when these tenants move into the house, causing resources to be insufficient for everyone. How will the landlord respond to this crisis? For example, will they reduce the number of offspring, like having twins reduced to a single child? Returning to my research question, we primarily explore how the presence of gulls affects plant reproductivity, with a particular focus on pod characteristics and seed production. Specifically, does the presence of gulls influence pod length and mass, as well as the number of seeds? Additionally, how do different levels of gull infestation impact the plant's reproductive output? These questions form the core of my research. You might wonder why I decided to do this research. There is a significant knowledge gap about how gulls affect their host plants, particularly in areas like pod length and mass. While previous studies note gulls' impact on seed production, detailed research on plant other aspects of reproductivity is lacking. There is also limited research on the interaction between Australian native plants and gulls, which I aim to address. We need to understand how plants allocate resources between growth and defense, crucial for their survival and trade-offs. This research will also explore how plants and insects interact, especially how plants adapt to parasitism. So how exactly I conduct my research? First, I carry out field work at Smith Lakes, where I selected 3 groups of acacia trees with different levels of gull infestation. These groups include heavily infested trees, where every branches had gulls, completely healthy trees with no gulls, and partially infested trees where some branches had gulls and others were unaffected. By comparing these groups, I aim to understand varying degrees of gulls infestation impact on host plants' growth and reproduction. For our data collection, we first randomly selected 5 branches from each tree that were in similar condition and had pods on every branches. From each branch, we collected 5 pods for measurement. For each pod, we measured its length, mass, and the number of seeds inside. After collecting the data, I performed statistical analysis. First, I used two ANOVA to compare pod length, mass, and seed count between healthy and infested trees, as well as between healthy and infested branches on the same tree, in order to determine whether there was significant difference between groups. Then I applied linear regression to examine the correlation between goal numbers and pod mass and length, assessing whether more goals lead to smaller pod mass and length. In our expected outcome, first, the two-way ANOVA analysis will reveal significant effect of presence and numbers on pod length, mass, and seed count. Second, linear regression will help us uncover correlation between goal numbers and pods, such as whether increased goal numbers lead to a decrease in pod length and mass. Finally, these findings will advance our understanding of plant resources, allocation patterns, and trade-off under parasitic stress, providing new perspective of new research. In conclusion, our study highlights the significant impact of gulls on plants' reproductive traits. Future research could explore long-term effects on host plants and further investigate the mechanism behind resource allocation and trade-offs under parasitic stress.